Thank you all of you for coming out this morning at 9:30 on a Sunday morning. We do not expect this to be the most filled up talk slot, but I do think that it's going to be worth at least the 45 minutes that you're sitting here listening to this, especially if you're not familiar with this field of research. So I'll kind of give you a bit of a background. As Chris said, so I started in cancer vaccines under Bernie Fox, one of the kind of founding members of CDI. I've been kind of in that space for quite some time. And so I think about a lot of this from kind of the cancer immunospace, but we're going to talk more general immunology today. And I kind of get into why I think the technologies that CDI is helping provide are really important. And that I hope some of you end up doing some work with us in the future if you haven't already. So Seromics, what is Seromics? I don't even know if this is the word for it, but high throughput serology, right? Like, so what is the totality of the secreted antibody repertoire? We're not talking BCR, TCR sequencing, which is generally antigen blind. What is the actual universe of antigens that are bound by human secreted antibodies? This is essentially an unknown thing at this time, and we're calling it Seromics because the most common assay we do is serum. We get essentially identical results from plasma, but there's also secreted antibodies, obviously, in CSF and other bodily fluids. So why should we care about this? Well, the human body is essentially a walking immune system. About half of all of your nucleated cells are immune cells, and they're there to protect and keep everything else going on. So they're not the majority of the biomass. This is a really cool review where they tried to actually break down the number of things. So an immune cell is dramatically smaller than a skeletal myocyte, but by having its own unique nuclei, it's able to have an unique adaptive immune receptor. And so the reason that we have so many immune cells, particularly the T cells and the B cells, is each one of those and their variable region is an opportunity for a dice roll on some sort of random shape that might match foreign pathogens and invaders. And there's a lot of data that's stored in that, right? So there's perhaps as many as 1 trillion B cells in each of our bodies, perhaps 100 billion of those being unique clones of either naive or activated memory cells. And what are they doing? What is the universe of stuff that's going on there? There's a lot of complexity that we as immunologists have still really to unravel. So the purpose for this diversity, right, is there's a whole universe of insults, pretty much everything that the 1.5 billion or a couple billion years of evolutionary life on Earth can throw at us. Your adaptive immune system is trying to stay ahead of viruses and bacteria. It's really all possible antigens on Earth is what that randomness is trying to anticipate. And so when it's doing its job, it's fighting bacteria, it's fighting pathogens, it's recognizing foods as being harmless. But really when there are errors to that is when there seem to be kind of more important things on immunologic chronic disease that most of our clients come to us with, which is how do these responses, when they're not going right, they start to cause diseases when they react with the human proteome, right? So even though the intent of the immune system is to bind all of these foreign things, every single one of us in this room is walking around with high affinity autoantibodies, and not all of these are necessarily disease causing. And so a lot of what we try to do at CDI is try to figure out what is it that these antibodies are doing and help kind of point people's research in new directions. And so why should you care about this? So to kind of get that example across, I'm just going to show one really clear example where a very simple mechanism in the autoantibody serum, a very simple mistake, causes a very profound and important problem. So probably the simplest way to think about this is just via unintentional mimicry. A pathogen epitope looks very similar to an autologous epitope, causes a bad cross-reaction. So this was shown really clearly by groups at Stanford and UCSF, where they showed in the case of multiple sclerosis, what is typically seen as a very complex chronic disease is potentially caused by a very simple upstream mechanism. What they showed is there's a particular epitope of Epstein-Barr virus, EBNA1 protein, that cross-reacts with a human myelin protein called glial cam, and it's just this one epitope, and they're able to recapitulate that in mice. You inject the EBNA into the mouse, it makes the mouse cells go and attack the neurons, and there's just enough homology there that preserves. And they were able to identify reactions to this epitope in about 1/4 of all multiple sclerosis cases. And so this one thing might be the cause of all of those downstream effects. And so being able to see that type of effect, I think, is a really profound thing. And they used it with a high throughput, total unbiased virome assay called VirScan, and they identified that specific glial cam epitope on one of our HuProt protein microarrays, which we'll get to in a minute. So this is really important, right? This is a potential novel biomarker for MS to recognize people before they're diagnosed, and it is also a druggable target. So the same way that you can make a cancer vaccine to attack something, there are whole groups of immunologists right now getting spun up on tolerogenic vaccines. And so what you might need to do to cure MS is build a perfectly tolerogenic therapy about this thing. You need to either eliminate those B cell clones, maybe do a CAR Treg to shut down the response, but you now have a target to go after. And kind of the whole point of this talk is I'm imagining there are tens of thousands of things out there like this that have to do with things as severe as MS and as minor as why your knees get creaky in old age. And if we could understand when and how and where this type of stuff is happening better, I think we could get a much better handle on how to better age and heal our immune systems when things go awry. So the reason I'm up here talking to you today is because all of these people make those HuProt arrays that part of that study was done with. The arrays are quite difficult to print, so Shuang Liu, our lead print engineer in the middle, has a PhD in physics. It's not a trivial thing to do these, and so HuProt is essentially the majority of the human proteome on a nitrocellulose slide, so over 21,000 full-length human proteins. And this is the result of about 15 years of work by our scientific co-founders. So originally out of Johns Hopkins with Seth Blackshaw, Hung Zhu, and Jeff Buca, started CDI about 15 years ago now. And the original thought was this is going to be a cross-reactivity screening tool to see whether or not a monoclonal antibody was actually monospecific. So the goal was to build really good monoclonal reagents. It was part of something called the NIH Protein Capture Reagents Program, where they built really nice monospecific array-validated proteins or array-validated antibodies that were against human transcription factors. But what they essentially started finding out as time progressed is that the majority of our clients were immunologists like myself. And so I had actually worked with Invitrogen's protoarrays, monitoring cancer vaccine responses back when I was in grad school. Turns out so Mike Snyder at Stanford is famous for inventing the original protoarray. It's actually his postdoc Hung Zhu that did most of that work. And so Hung became faculty at Hopkins. And so same person that invented the protoarray invented the CDI array. And so just to be clear, so we are a proteomics company that makes proteins. Companies like Olink and SomaLogic and Alamar, they build reagents that detect proteins. We build proteins to detect other things, most commonly serum antibodies, which is primarily the discussion of this talk. So our tools, the HuProt platform, is the most cited, the longest available thing that we have. You can use it really for two different things. You can have one special target, like the original monoclonal project. You can figure out if you have an antibody, you don't know what it's binding to. We can screen against the entirety of the human proteome. Or you can take much more complex samples and try to see what's going on across wider cohorts. And that's what we do on the Seromics side. So as kind of mentioned, so we have full-length human proteins. These are all GST tagged, made in yeast. That GST tag helps solubilize membrane proteins, but they're not membrane embedded. So you imagine the epitopes might be not fully naturally folded on the standard HuProt. That's why we're getting also into a separate platform called VIRD. And VIRD is a membrane embedded HSV1 virion that we swap out one of the membrane proteins with a human protein. Right now we have essentially the majority of the non-odorant GPCRs and about 100 immune receptors on the surface. So those are it's an actual membrane embedded array. And then we have a separate platform. We're the first company to commercialize phage-display immunoprecipitation sequencing. What these are long bacteriophage libraries. So we've got 49 to 62 mer peptides, hundreds of thousands of them covering the entirety of the human proteome, the entirety of the mouse proteome, and the entirety of the viral proteome. And we'll talk about that a little bit more. And so it's a more linear epitope structure than the full-length human proteins on HuProt. So how we go about actually doing these assays is, these are complex platforms. There's a lot of things that goes into them. We try to be really transparent about our QC processes, try to show that we actually make the proteins that we claim to make, that we're actually able to stain them correctly and give just kind of a stack of output materials demonstrating that. So this is what a single HuProt microarray looks like. This is actually a pseudo-color image reconstructed from the array data. So we're kind of seeing little dots light up. Those are the reactions for this individual patient's array. We're generally staining 2 channels at a time. And this is what a really good one looks like. And we try to demonstrate that with every array delivered. So if there's a bad artifact, we'll restain it. As far as the collection itself, there was a capstone Nature Methods paper as part of the NIH project, which is how we demonstrate the sequences are what we think they are. And then we produce all the proteins individually, and every array lot we stain for anti-GST to prove that the proteins are successfully made and are there. And then with every individual project, we have every spot printed in duplicate. And so the alignment and the correlation of those spots helps us confirm that we're extracting the data correctly. This is now what the top end of this particular array looks like. So what are the spots that are actually lighting up? So this is a patient with scleroderma. So they've got anti-centromere B proteins, but you'll note, and this is a very common thing, they have a canonical autoimmune disease. This is a canonical autoantigen that they're screaming positive for on HuProt. It's not the hottest thing on their array. This is the same for lupus patients. This is the same for almost everyone you look at. Everyone has high affinity autoantibodies that arise from some event that has occurred throughout our lives. What is the purpose of this? Not really known. And so kind of mentioned, so there's a log z-score of what's highest, the physical image of what's lighting up, the background secondary only stain, proving that this isn't a false positive induced by the secondary, and then the anti-GS stain from the actual array lot, proving that all the proteins got made. And so we try to be pretty simple and transparent with our statistics, and then let clients do more clever things with it after the fact. So additionally, as I mentioned, we have our PhIP-Seq platform. Again, this is complex libraries of bacteriophages. What's really nice is the DNA that creates that peptide is actually there in the assay, which is great for detection, which is great for validation that what you thought you made is what you actually made. We then do barcoding and sequencing to get specific readouts. And we kind of have a similar process where we go through, we prove that the library was there with every sequencing run. We prove that all of your samples have good high counts. And then we use an algorithm called EdgeR to call specific hits versus decoy background. And so this is just what a raw data table from a VirScan assay looks like. And it's a little small, but what you have over here are a lot of annotations, the start-stop position of the peptide, what protein and what virus it's from, and then the specific amino acid sequence, right? So this is from a spreadsheet that's 480,000 rows long. And this is run alongside about 100x coverage of the background library. And what you'll see is there's just, random signals that are going all over the place because you can observe the background library in pretty much every sample. But when you get a reaction, so this one says 3,600, and it's a lot more than the 30, 40, 20, 18 that are the patients that are negative. And so when you get a real signal, it kind of smacks you in the face. And we use about 100x coverage, so about 40 million reads of the background library to each individual sample run, and then we use EdgeR to actually call those specific real reactions. And so this is a case control output of just some healthy old versus young people, so people like 60 to 90 versus people that are about age 5 to 16. And so what's most different between those two patients? And we just go through and call binary reactions, true, false. And what you see is there's a particular herpes virus peptide, which all the older people have been exposed to, that the children have not, that are negative in the beads only mock immunoprecipitation background. So again, this is a protein AG pull down. So it's a very IgG dominant assay. And then we give an output at both the peptide, the whole protein, and then a polyclonal signal. And what's really nice is because you have this peptide, it's not just that one peptide, you can look in the context of the whole protein. So each column is a patient, and each row is a different amino acid down this whole protein. And the peptides in this VirScan, at least, are only partially overlapping. And so you don't have a dual reaction, you have a single reaction, and this seems to be the epitope that is the immunogenic piece that these people are reacting to. And it's a quite clear thing that you can see. Here's an example of EBNA1 from EBV. We were talking about that earlier in that multiple sclerosis paper. This is actually the literal library that was run by that. So we've licensed it from that lab. And so now you can actually see that this is more of a dual reactive. The epitope's in that overlap region, and you can see that coming up in multiple people. We see that both the adults and the children have been exposed to Epstein-Barr virus, which is very interesting. Over here on the right are triplicate controls that we always run. We run a human pool plasma, and you take any pool of human plasma, you're going to get EBV reactions in it, so we see that. And then we also have an anti-GFAP, which is well published on as a kind of a control polyclonal that seems to come up regularly reactive in the VirScan assay. So these are the kind of the way that the data comes out. This is the way that it looks at it, but why should you care about this? And I think the reason you should care about this, there's some really profound examples of the human serome or the antibody repertoire, both autoantibody and antiviral, having a really deep control over our health that is independent of genomics. And one of those places that we might want to look for places like neurodegenerative disease where we currently don't have good explanations for what's going on. So there's quite a few publications related to this. It's still a relatively small field. This is as of last summer. I think we have about 240 total citations in the literature right now, citing primarily our HuProt arrays. And what is it that autoantibodies can do? They can do lots of different things. They can have a very simple molecular mimic response. Like I said earlier, we've got a natural antipathogen reaction, which is a good thing, that cross-reacts with a native peptide. That could be a bad thing. It can also be a good thing that antibodies are destroying things. So in the case of tumor antigens, you want autoantibodies against tumor-associated antigens. But there's kind of a deeper weirdness to this that really is part of what convolutes the data. And that's that you can have naturally occurring drug-like autoantibodies that naturally eliminate or knock out or alter immune pathways. Jean-Laurent Casanova has done a lot of work on this. He just had a review that came out in one of the Nature family journals last week. Would encourage you to dig up. I can maybe try to find it after the talk. But so I'm just going to show a couple of examples of things that you can see in this type of data. So these are patients that were treated with anti-PD-1, anti-CTLA-4, so ipilimumab, nivolumab, or the combination thereof. And this is by Iman Osman's group at NYU. And they're able to show with a pre-treatment HuProt microarray signature, there are things that the patients have autoantibodies to that predict their outcomes to canonical tumor immunotherapies. These are the validation cohorts of several hundred patients, as well as a waterfall plot on the immune-related adverse events. And so there's something going on here that you can see. Like, is it the tumor, the state of the tumor just being mimicked and mirrored by the immune system, or is it something deeper? I honestly think that it's likely something deeper in many cases, but they didn't really get into and parse apart mechanism underlying the list. They just came up with a list. Another way that you can kind of define cohorts, this is some work that we did with Benjamin Bessie's group with Gustav Rousey, and we profiled a bunch of lung cancer patients on VirScan, showed that cytomegalovirus infection being active and having strong antibodies to that was correlated with senescence. So if you do spatial biology and see if you've got a hot tumor versus a cold tumor, the people with the colder tumors with the less active T cells had active CMV infections. And this has actually been known by tumor immunologists for quite some time. CMV is really good at hogging the immunologic space and distracting it from anti-tumor immune responses. We're able to show that in a very broad, unbiased manner using this powerful tool. And it's just kind of a cool thing to show. But this, to me, this is the part that's most interesting. Like the fact that some of us are probably in this room have immunologic knockout-like effects induced by our immune systems. And so Steve Holland, who's the clinical director of the NIAID, is really interested in this. And I would highly encourage you to take a picture of this page and go watch this lecture online. There's only a few dozen people in the room at that time, but one of them was Steve Rosenberg, who's one of the godfathers of cancer immunotherapy. Like, interesting people are interested in this. And what Professor Holland goes through and shows is very clear cases in infectious disease where certain people only get certain infections when their immune system has previously knocked out something like interferon gamma or interferon alpha or GM-CSF, things that we know do things, right? And that there's probably some background frequency of this in the general population, and perhaps this is something you should care about. Some of the places where it's been shown more clearly are things like air deficiency, so having the standard tolerogenic transcription factor in the thymus, air or APOSED, these kids get catastrophic autoimmunity. This is a paper showing that it's not air, it's a separate air-independent thing that causes an air-like syndrome, and it recapitulates effects that are regularly observed in air, such as patients get anti-interferon pathway autoantibodies, and that this then affects the type of disease progression they have, such as whether or not they get type 1 diabetes. And this was all shown on HuProt arrays. So, it's kind of a general principle is that sometimes, you get an infection, the cytokine does its job, you clear it. But now we've got this weird wrench being thrown in there, which is that sometimes people have autoimmunity to cytokines. And that's really interesting. But, there's only so many cytokines. There's only so many things to check. Like, how common is this really? But what I'd like you to kind of expand your head on a little bit is there's a lot more on the surface of cells, and there's a lot more signaling molecules out there than there are cytokines, right? There's 3,500 surface proteins, and this is essentially unexplored. And we regularly see autoantibodies and HuProt arrays in regular people, unrelated to disease, to circulatory and membrane proteins. What are those doing? They're not always doing something. Sometimes they bind in a harmless region, they don't do anything. Sometimes they bind a functional region. Sometimes they actually reduce the clearance. There's literature that you can actually have like a cytokine lifetime extending signal from autoantibodies. So it's the first step is kind of the discovery process, but then there's a lot of deep immunobiology going on that we still haven't really figured out a way to parse at scale as to what these things are really doing after the fact. So what do we know about the human serome? So this is kind of my current best assessment of it. Nobody's really done a proper full life history study, but kind of piecing things together, and I'll show a specific example of each of these, is that all of us have autoantibodies, that most of it probably arises earlier in our life, like when we're very, very young, when we're first getting a lot of our natural background infections to viruses and vaccines, kind of an antigenic first sin type thing, that it's generally stable across time, that the autoantibody serum is more unique than the antiviral serum, but you'll still get new responses with time, but not everything causes that, and that this is a very genetically independent thing that performs very differently among identical twins. So everyone has autoantibodies. So I'm just going to tell you to take my word for it at the moment, but depending on where you set thresholds, 50 to 400 reactions is about what we see on HuProt arrays. On PhIP-Seq with the HuScan platform, it's about 500 to 6,000. It really depends on where you set the thresholds, right? So with these discovery platforms, we're usually doing a gut shot, single dilution, 1 to 1,000. We're not doing titers. And so to really get a better sense of the scale of this, like we're going to need to run more samples, we're going to need to run titers, but this is kind of my general estimate. So most of the serome arises earlier in life, and there's not a lot of people doing life course studies. So if you have such a cohort and you'd like to do one with us, we'd love to see it. But one of the examples in the literature that mirrors stuff that we've seen with people of different age groups, this is by Josh LeBaire's group at the University of Arizona. And what they've gone through and done is just profiled people at different ages and saw on their microarrays how many people, or how many autoantibodies are they counting. And that they see that the very young children have fewer than the older people. And that's about the best that exists on this. Like ideally, you'd like to profile people across a time course, but that hasn't really been done so much yet. The autoantibody serum is generally stable across times. This is done on HuProt arrays, so you stay in the array with serum at 1 to 1,000. You come back with a fluorescent secondary against IgG, and then you scan it on an array scanner and see what the output data is. And this is now just a hierarchical clustering of all the HuProt data, and it's a little hard to see, but essentially we ran healthy women in middle age 1 year apart, so like age 55 to 56, and we ran their HuProt arrays, and they look almost identical. And what this is showing is that everyone pairs with themselves after 1 year, and they're not randomly pairing with someone else. So there's a very unique fingerprint in there, and I'll show a bit better examples of this in a couple of slides. So the autoantibody serum is more unique than the antiviral serum. So this is work that we did with Shan down at Emory, really early in the pandemic with kids that had MIS-C syndrome that were visiting his clinic. And we ran phip-Seq, both HuScan and VirScan on these samples, so a whole human proteome, whole human virome. And this is really early in the pandemic, so COVID-19 wasn't actually in the collection. This is just pre-pandemic SARS. So each column is a patient. Each row is a different epitope, a whole scan of the spike protein from SARS-1. And over here in the red column, we have healthy children that have not been vaccinated because it's early in the pandemic. They have no reaction. Whereas the kids that are either in the acute or convalescent after they've recovered a little bit are starting to see some of these shared epitopes. And the kids with MIS-C, same thing. Over on the right, same with nucleocapsid. So we see early infection, late infection. You start to see more cross-reactive anti-N reactions come up the longer that you've allowed the immune system to develop on that. And, you know, there's just enough homology. So I was really excited early in the pandemic. It's like, wow, there's that much homology between SARS-1 and COVID-19, but our vaccines are going to be really cross-protective. And it's kind of part of why I hope that, you know, coronavirus isn't as bad for us now that we've all been exposed to it in some manner. There's a lot of cross-reactivity. The way that the ACE receptor has to latch on to spike is kind of evolutionarily forced. And so hopefully this type of cross-reactivity continues to exist. But what about the rest of their profiles? So these are now Pearson correlation plots that are summarizing the entirety of the data. So there's about 100,000 data points on the VirScan side per sample. There's about 250,000 on the human side. And so it's taking what is the entirety of their autoantibody profile or the entirety of their antiviral profile. So this across all possible viruses, all possible human proteins, what are people reacting to? Well, so bigger circle means higher correlation. And these are all paired time points. So people that are the same people during the acute and convalescent stage. And you see those lining up. So people look like themselves early and late infection against all viruses and against all autoantigens, but they don't really look like each other. So everyone's autoantibody profile in this, all these children have rather unique autoantibody profiles. But over on the viral side, we see bigger circles, more correlations. So because we're all exposed to the same viruses, the same vaccines, they're all supposed to be foreign, you're supposed to be able to recognize most anything over here. That's what's more shared, but still relatively unique between individuals. But the way that you'd really want to prove this would be with identical twin studies. And so that's what we did with Mario Fauci's group at Twins UK. We got 20 sets of lean and obese twin pairs across about 10 to 15 year time gaps. So age about 55 to 70. And these are all identical monozygotic twin pairs. And we're running all of our Seromics platforms. So I'm going to show the phip-Seq data first. And so this is the same type of plot that I was just showing before, except now we've got 2 time points and twin pairs. So there's kind of like groups of 4 that we're cross-comparing. And so this is twin group 8, the first individual when they're 55, when they're 70. Twin group 8, this person's identical twin, age 55 and 70. And what you see down this whole plot is this really beautiful stair-step pattern where everyone looks like themselves a decade later. And this is with autoantibodies, by the way. The viral plot looks very similar, but the autoantibody profile of people is stable across more than a decade, and it looks almost nothing like their identical twin. And so the types of errors that the immune system are making are essentially genetically independent and unique. And we all know that, right? The immune system is an adaptive, intelligence, independently arising system, and there are some biases and things that get shared amongst twins, but most of that is still happening independently. And so just to kind of show what the microarray data looks like for the same population, now we're just looking at one of those sets of 4 samples. So it's twin 1 on the left, twin 2 on the right. So these are identical twins, time point A, time point B, and there's a decade, right, between these samples, and this is their HuProt signal. And so what you see is a really tight correlation down the diagonal of the samples reproducing. So I want it, unfortunately, we've got these green dots in there that are more control proteins, but there's about 100 blue dots up here too, which are the real kind of autoantigen reactions. And it's very stable across time. There's a bit of noise down here at the bottom that kind of makes this not quite look like replicates. Like if I just ran 2 replicate samples, it'd be really tight on the bottom end too. So there's a bit of noise in the really weak signals, but for the strong stuff, that's very, very well preserved. And there's a lot of them that have essentially no change in their autoantibody serome across a decade, which I really think is a rather profound observation. But that's not the case for everybody. So when you give cancer vaccines to people, so we do some studies pre-post cancer vaccination, you see effects like this, where you see this whole kind of smear of novel autoimmunity that gets induced, but this doesn't always happen. So across 10 years sometimes it doesn't happen. Here it happened. What triggered that? Did this person have surgery? Did they get a bad infection? Not really known, right? But some sort of thing caused this event. We see events like that with cancer vaccines. We did run some people pre-post the Moderna, Pfizer, and J&J vaccines early in the pandemic, just 3 or 4 samples, didn't move the needle. People look the same pre-post. You give somebody ipilimumab or nivolumab, these kind of sledgehammer checkpoint immunotherapies that cause a lot of immune-related adverse events. Most of the patients don't get an effect like this. It's mostly stable, which is very interesting. And so this is now a really good summary figure that a collaborator of ours, Tim O'Donnell, put together across this. And I think this is the kind of the punchline of the talk and where we'll start wrapping up. But this is now summary data on that same twins cohort for all 3 platforms. So we looked at essentially all possible viruses, so 500,000 viral peptides. We looked at the entire human proteome on the HuScan assay, about 700,000 human peptides, and then the 21,000 human protein microarray. And this is now comparing the similarity of 2 samples by Pearson correlation. And over here on the right side, just everyone compared to themselves after a decade. And so what you see is on the VirScan, the HuScan, and the HuProt arrays, everyone's got a pretty tight correlation with their own samples to almost all the signals across 10 to 15 years. And they look very different from the different individuals. But kind of most strikingly is they're also looking very different from their identical twins. What's nice is by putting the data together this way, you do see that there is a difference, and everyone does look like their twin slightly more than the rest of the population, but in general, that's an independent thing. We also see what I was talking about earlier with the Emory samples, where if you look at the VirScan data, that's where the different family correlations are the highest. So we have more shared immune repertoire with our neighbors for things that are general viruses and things circulating in the population. But the autoantibodies are profoundly unique. And there's been some cases, as I showed with the MS paper, that really means something. And I think that kind of us as a field, we don't really know what all of it means yet. And so kind of what we're trying to do at CDI is provide access to this data and try to kind of open up more of this type of science and research to more groups. And so kind of with that, I'll kind of wrap up the talk. And thank you all for coming on an early Sunday morning. And we've got the room for like another 20 minutes. So happy to talk more.
All the 21,000 proteins in a HuProt is confirmation as wild type when you're expressing it. That's point number one.
Well, start with point number one, and then we'll go to that. So we cannot check all 21,000 proteins. So the most important part is there is a three-dimensional GST tag at the N-terminus of every protein. So there's a protein that has to be folded for us to purify it in the first place. So that helps, but that does not assure, of course, that all possible proteins are folded. We've done kinase autophosphorylation assays that show that a lot of the kinases will autophosphorylate and are in a native state on array, and that we can ablate that with urea. And probably the best demonstration is we've got about 200 peer-reviewed citations of people being able to follow up and prove that what they found on a HuProt array actually mattered. But the majority of the collection, the entirety of the collection, we can obviously not prove is folded. And in particular, the membrane proteins, we imagine, is more of an issue.
The second question is more with the performance of the assay. Is there a publication or internal data that shows if you have compared post-COVID infection autoantibodies of the same individual changing or not changing? I think you mentioned about 2-3 samples that you tested. Is there a larger study?
Yeah, so that was just a handful of patients that were pre-post vaccine. That whole study that I was showing with Emory kids was pre-post COVID vaccination. And so they weren't getting a lot of novel autoantibodies. We do have a few groups that have done rather substantial long COVID studies with us, but I don't think the majority of those larger cohorts are published yet. So yeah, I wouldn't know all the pairings in those because some of those we ran blinded. But, yeah.
Sorry, I came in late, so I might have missed this, but do you look at, for example, like IgG1 versus IgG3? Is that something that you can do with this assay? You may have said it.
No, I didn't say it, but I will get into it a little bit here. So that's actually part of why I'm here at CDI. So when I finished my cancer vaccine PhD, our CEO at the time, one of the founders, Ignacio Pino, was searching on Google about antibody isotype ratios that have to do with T-cell responses. And my PhD thesis was the top Google result. So we talked and I joined CDI. We don't do that much yet, but IgG does not exist. It is composed of all these subtypes. And I think deconvoluting that data is really important. And so actually my kind of few punchline figures is, so I've done that. This is not published yet, but so this is one person's serum stained for IgG1, 2, 3, 4, M, A, E, and D. And this is quantile normalized, so it's kind of artificially inflated. The IgG4 signals, I should probably redo it without that. And then essentially each dot is a protein on a HuProt microarray. And I've just essentially thrown it into a Visney algorithm like you'd use for flow cytometry. And what you see are these kind of little islands and patterns that are forming among the different isotypes. And if you kind of go through and kind of play with gating that, like with flow cytometry, this is now the same type of data with like a healthy kid versus a child with an autoimmune disease. And now it's kind of all of them against each other. And you can kind of start going through weird, I dumped the data in FlowJo and was playing with gating strategies. And we haven't really dove into it super deeply, but like, so this is a case of a child with lupus. And this is IgG2, IgG1. And what I kind of found was the canonical lupus autoantigens are popping up in the IgG1, IgG2 subcompartment. And there are other things that are known associated with lupus that are proteins that are enriched in that region. And, you know, you can zoom in and further gate and play with like IgA2, IgE negative, and I don't know where to go. So part of the reason we're not doing a ton of this right now is there's not actually that good of really good secondary reagents for this stuff. We're in the process of developing some of them ourselves. We'd actually like to be able to do capture pull downs that are pan-isotype for PhIP-Seq. And so that's something I hope we can bring to you guys in the next year or two, but it's kind of on my skunk works list. So it's kind of convenient you asked that question. Thank you. Yeah.
Hi, I was looking at your website and you also have mouse scan. So like when we kind of use transgenic models to kind of run some autoimmune disease, in that studies, like if we have to detect autoantibodies, is that this assay will kind of support that?
Yeah, so there's a couple of different ways. So I think part of why this field is still underappreciated is because there are not very good preclinical tools. Like the incentive to build a mouse proteome microarray is not terribly high for industry, and it's a very expensive and challenging thing to do. So as mentioned, we do have a whole mouse proteome PhIP-Seq assay. This is developed at a Joe DeRisi's group at the Chan Zuckerberg BioHub. They've got one publication on it. I'm about to submit a publication on it. We've run, a handful of customer samples. It's not really been used that much yet. But I think the way that you really establish the importance of the life history of how the serome behaves is going to be done first and best in mouse models. And we can run that today. It's the exact same assay, same outputs as the humans, and the mice have autoantibodies. I unfortunately cut out my mouse scan slides, but transgenic mice look kind of like the identical twins. They look more similar than the human identical twins because they live in the same cage with a very similar population. But the autoantibodies are still pretty hyper variable. It's interesting.
Hi, Tian Fu from University of Houston. Yeah, so I just want to know the quantitative capability of this one and the standardization of the protocols, across different labs, and if they run, can they have like a reproducible results or so this kind of thing?
I think that is by far our biggest challenge as a company right now, is because we remake the proteins about every year, which slightly scrambles their concentrations. The clones reproduce about the same. You'll get top-end stuff for individuals that we see preserving across years and across lots, but that's in our hands where we've got SOPs and we're using the same reagents and we're doing the exact same thing with the same technician's hands every time. Like really dialing it in and getting the exact same results is super hard. And so DNA and RNA microarrays ran into a lot of problems with this. And people try to do all these clever normalization schemes. What I've kind of evolved to is just a really clean system where you stain a background array of the secondary, you stain your patient slide, and you just do a log2 transform. You don't do any quantile normalization, and then you just look in deviations above that secondary only within the individual patient. And I'm calling reactivities and pools, and I'm getting good reproducibility, but I don't think it's entirely established what the best way to do this across labs is. And we'd love all of your help in thinking about that. We're thinking about setting up an actual Seromics meeting to try to help get into some of the standardization stuff, because there's really low-hanging fruit, like making sure everybody uses the same reagents, that we could solve before you get into other things like normalization.
All right. So, but you don't have like a standards or so that you can sort of get a. So this is still using fluorescent intensity, right? Yeah. Okay.
Yeah, so it's a semi-quantitative platform, right? So we have like different ladders of IgG printed and an anti-IgG printed where you can see the controls behaving the same way across all of them. But really, there is quantitative data here on both platforms, but all the proteins are printed as purified. So they're different concentrations. And then the antibodies are working on log scales of titers. And there is so much going on that I've almost moved to a binary yes, no hit call for things. And that seems to reproduce a lot better across studies than trying to be really quantitative with it.
Can you think about including like a serum pool to normalize across different plots?
So that's a challenge for Rich Boykin, because we are running serum pools with every array lot, and we're running a pool triplicate for every PhIP-Seq lot. So we have that control included in pretty much every lot and study that we run for the last several years, but we need a way of normalizing to it. So that's that man's job over there.
I'm just curious about the biology. For autoantibody, are they like a yes and no kind of response, or they have a gradient and a different level of concentration?
They totally have gradients and things of different concentrations. And so where I try to move myself towards the things that reproduce most obviously, the really strong signals. What's really interesting, it's one of the examples that I used. There's a Nature paper right here where they actually go through and they validate that these are function modifying interferon autoantibodies. And there's HuProt signals on gene-pick scanners just like we use. And the range of these signal, the dynamic range, it's 16-bit. So it's up to 2 to the 16 or 65,500. And so I usually don't even start caring about things until it's like in the 400, 500 range or something. And they're chasing and finding real things that are just minuscule responses. And we see that quite often where there's really small, highly significant signal differences between groups. I don't know whether they're important or not. I think sometimes it's like different proteins in the serum acting as a sink that's just altering the distribution of the signal, because like these are complex samples. It's not just antibodies that are in there. But I think that's a very good question. I don't think we as a field have a good answer for that yet.
I think someone already discussed about how you normalize your data. I think a big concern always like the difference across slides or across array. So I think, yeah, I think we would like to know more about how you do that. And another question about the isotype, I think someone already asked. So do you do like IgG, like a IgA, IgE besides?
Yeah, my favorite combination right now is total IgG, total IgA. If you have a secondary, we can run it. So microarrays are really flexible. And if you've got a reagent you really like, we can stain them with anything. It's just that there's not reagents that I really trust for, say, IgG2 right now or something like that. But if you have something, we're happy to run it. And I'm sorry, I forgot the first half of your question.
Normalization.
Yes. So historically, a lot of people have done quantile range normalization. But what's kind of hard about that is it artificially expands or compresses the number of total reactions based on what's there. And so it's really useful for tidying up and finding outliers, but it kind of artificially does. So one of the things that I hate the most about my multi-isotype that I did was back when I did this, I was putting everything through quantile. And this patient does, there's like 5 that are probably positive on IgG4. And we're seeing the whole thing light up. And that's because I pushed all of it through quantile and it's artificially boosting. So I actually don't like that. Like where I've really gone is just a log transform of the raw data, paying attention to where the secondary is performing, and then being really tight with your staining and scanning conditions. But ultimately, we are going to need better normalization than that. But what we found is that there's nobody that have really written normalization algorithms specifically for our data types, either this or PhIP-Seq, that seem to do a really good job of cross-comparing studies. But if we had a central database or place to start putting all of that, I think we might start learning a little bit faster. What we're finding is most labs are coming up with their own normalization methods. Almost everybody that puts out a Seromics paper is doing some sort of different custom analysis that they invented to go after whatever it is they find interesting. And I don't think any of us really have this worked out yet. So, thank you all for coming. I mean, we got a booth and happy to talk to any of you the rest of the day. So, thank you.